So our next speaker is Olivier Pourquié from Harvard Medical School, Harvard University, not medical school, I think. Okay, anyways, from overseas, and he will be telling us about. So Olivier is going to talk about physical principles. That's what he put in his title. So physical principles of what? Well, underlying patterning of the vertebrate body axis. Thanks, Olivier.
Okay, so thanks, Thomas, for the invitation, and I apologize for the pompous title. That's the only way I found to not to tell what I was going to talk about, because I wasn't sure. So, so today I want to talk about the process whereby the body axis is formed, which is called elongation in vertebrates. And so I just want to start to introduce this in very simple terms, and just remind you that the way the axis of vertebrates is built, and that might be different in invertebrates, especially flies, for instance, is first by laying the anteriormost structures, the head, and then adding progressively the posterior structure from a growth zone, which in amniotes is called the primitive streak, and that's what we heard from Jérôme earlier, which morphs later into the tail bud. And so this growth zone produces cells which contribute to increasing the volume of the embryo, and that become patterned into this stippled area, so what I call here patterning zone, and eventually they acquire some kind of morphology, like for instance shown here for the segments, which in vertebrates are called somites. And so this process is ongoing, and so the progressive addition of tissue is called the axis elongation, and that's the process whereby the posterior body of vertebrates is built. And when I mean posterior body, I mean pretty much anything down to the cervical region. And so the way it looks like in real life is illustrated here. This is a chicken embryo at 2 days of development. So one difference compared to here is that this has these axial organs in the middle, which include the neural tube that will form the spinal cord and the notochord. And it has this clear bilateral symmetry that you can see here. And so the axial organs are flanked by a tissue which is called the paraxial mesoderm, which is represented in red, that forms these strips of mesenchyme in the posterior part of the embryo that forms the embryonic segment. So the paraxial mesoderm, which I'll discuss to some extent in this talk, arises from gastrulation, first at the level of the primitive streak and then in the tail bud. Then it forms the presomatic mesoderm and then the somite. And so this is just to give you a more dynamic view of how elongation proceeds in the embryo. This is a time-lapse movie of a chicken embryo where you can see the ongoing elongation. Oops. You see here, this would be the growth zone, and you see here the presomatic mesoderm and the forming somite. So you see this is a very dynamic process that goes on until the total number of segments is formed in the animal. And so here what I want to show you is a movie that illustrates the fate of the cells of the growth zone, so the precursors of the paraxial mesoderm at this stage. And so the cells have been labeled with by electroporating a fluorescent protein, which is going to label the nuclei. And what you see here is the primitive streak. So this is the growth zone. And you see that it moves posteriorly. And while it moves posteriorly, it releases cells that will form the presomatic mesoderm and eventually form the embryonic segment. So this is a very dynamic process, which in fact resembles a bit the way a plant meristem would work, a shoot meristem, for instance. And okay, I'm struggling with. Yeah. Okay. Just want to show you this again. So you see here these cells, and so there is a progressive addition of cells that essentially is going to give rise to progressively the more posterior structures. And so there are several different ways you could explain such a behavior. And so, for instance, you could imagine that the stem zone is dragging the embryo posteriorly, and that's explaining the elongation of the embryo. That's one hypothesis which has been privileged for some time. You could also imagine that the tissue is somehow pushing posteriorly, and so many different kinds of possibilities. And to test between these different ideas, what Bertrand Benasseraf did when he was a postdoc in the lab was to perform ablations of different regions of the embryo, for instance, the axial region, as shown here, or the paraxial mesoderm, so in this case, bilateral ablation. And what he found is that when he removes the axial region, that doesn't prevent the elongation of the embryo, as you can follow here with this red line. But when he performed an ablation of a bilateral ablation of the posterior presomatic mesoderm, that essentially stalled the process of elongation, at least on a short time scale. And so what this was suggesting is that this region here of the posterior presomatic mesoderm plays an important role in the control of elongation. And so we had no idea how this might work, and started to investigate in detail what happens in this region, first at the cellular level. And so at this time, we teamed up with Paul François, who was starting his lab. No, I guess he was still with Eric Sigia in New York. And so what Paul did is to do an analysis of, to analyze the behavior of the cells of the presomatic mesoderm, so in the same type of movies that I just showed you. And what he found is when he looked at the motility of the cells, if you compare to the presomatic mesoderm, which is shown here, there is a high level of motility in the posterior region, so the region that controls elongation, which progressively decreases anteriorly. And furthermore, what he found is that there is a striking gradient of directionality of the cells. That is, that the more posterior the tracks of the cells in the presomatic mesoderm, the more the longer they are, and the more strictly posteriorly oriented they are, which was suggesting some sort of hypothesis where you could imagine that the cells in the posterior region tend to move faster posteriorly than the cells in the anterior region, and that could explain the process of elongation. But in fact, what we did next was to also measure the displacement of fibronectin, which was labeled with an antibody. And so we compared the tracks of the labeled fibronectin to the tracks of the cells, and subtracted the tracks of the fibronectin, which in fact reflects the deformation of the tissue, to the tracks of the cells. And then what we found is that we still have a gradient of motility across the tissue, but we completely lose the directionality component. Meaning that, in fact, in this tissue, if we look at the movement of the cells, what we have is a gradient of random cell motility, which would be akin to cell diffusion. And so this gradient of random cell motility was parallel to a gradient of signaling of the FGF, the fibroblast growth factor pathway, which you can see here, which peaks in the tail bud and decreases progressively as you move along the presomatic mesoderm. This is phosphorylated ERK, which is expressed in response to FGF signaling, which also shows this gradient. And with Marie-Claire Delphini, we had shown that this gradient controls the motility of the cells in the presomatic mesoderm. So if you activate the pathway, you get cells that are more motile, as shown in red, and if you decrease it with a dominant negative downstream effector, then you decrease the motility of the cells, which was somehow suggesting that FGF signaling in this context might act as an effective temperature controlling the random motility of the cells, the diffusion of the cells. And so also what we had found is that this gradient of FGF signaling is not established by a classical source sink model, but by a different mechanism wherein only the cells in the progenitor region, in this growth zone that I mentioned earlier on, actively transcribe the ligand that establishes the gradient, the FGF8. And when the cells enter the presomatic mesoderm, so where they will become actively motile, they stop transcribing this factor. And you can see this by using an intronic probe that only labels active transcription, which you see is restricted to the growth zone and not in the presemitic mesoderm. And then what will happen is that as the embryo continues to elongate, these cells remain at the same axial position, but its content, messenger RNA content, will progressively decay. And this mechanism will result in the formation of a gradient by RNA decay, which will accompany the elongation movement of the embryo. And what you see here, for instance, is the expression of the messenger RNA coding for this gene. So you see the difference between the transcription and the messenger RNA, meaning that here the cells they still express the mRNA, but they do not transcribe it anymore. And so the reason why I'm mentioning this is because this will endow the cells with intrinsic capacity or a defined level of motility when they will enter the tissue. And finally, the last component ingredient of the system is a gradient of density or a confinement of the posterior presemitic mesoderm within regions of high cell density. So what Bertrand did is to analyze the cell density in the posterior region of the embryo, and here you see two stages, 13 somites and 15 somites in chicken embryos, where the high density is shown in brown and the lower density is shown in blue, and you can see a clear gradient of density along the presemitic mesoderm as you move towards the somitic region here, and you can see also that the tissue is flanked by regions with higher density, like these are the axial organs, and this is the lateral plate. But clearly, the presemitic mesoderm, where you have this high motility behavior, is of much lower density. And so together, that led us to propose a model wherein this FGF acts as an effective temperature that controls cell diffusion along the presemitic mesoderm, and due to the confinement that I just illustrated, the system will generate a pressure that will be preferentially oriented posteriorly, and which will ultimately generate a push triggering the posterior elongation. So we've been modeling this behavior. I'm just showing you part of this from a cell-centric model that was worked out by Ido Regev in collaboration with Karen Gevorkian, who's here, and Mahadevan. And so the idea here is that this gradient of cell diffusion along the presemitic mesoderm pushes a wall and can generate a pressure in the way that I just illustrated. So now we wanted to start to try to validate this model, which is a bit counterintuitive for biologists. And so what we did, and this is the work from a PhD student, Arthur Michaud, who was co-supervised by Karen Gevorkian, who's now in Curie, but was in the lab for a few years before, and myself. And so what Arthur did is to try to deconstruct the system. And for that, he microdissected the presemitic mesoderm. Here you see a section of an embryo at this level. So this is this tissue here, and which forms these long mesenchymal strips of about 1 millimeter, a bit more in length. And he engineered a system of micropatterns, which are PDMS gutters of a width which approximately corresponds to the presemitic mesoderm, and in which he could introduce the presemitic mesoderm and look at their behavior. So in this way, they're put in the gutter, and then they're covered by a cover slip. And then he analyzed the behavior of this isolated presemitic mesoderm in these gutters, which reproduce the confinement. And as you can see here, they nicely elongate in these conditions, at least for some time. And you can measure the elongation of this isolated presemitic mesoderm. And so what this tells us is that indeed the presemitic mesoderm in isolation is able to generate these elongation movements. And then he looked at the role of FGF signaling in this context using inhibitors. And so what I'm showing you is PDO3, which is a MAP kinase inhibitor. The MAP kinase is downstream of FGF signaling in this context. And as you see here, the higher the dose of PDO3, the more you inhibit elongation. So with, you see that with 10 micromolar, you completely block elongation, while with 2 micromolar, you have a lower level of elongation. So which tells us that this elongation that we see in vitro is also FGF dependent. And then he looked at which region of the presemitic mesoderm is responsible for the elongation, and for that he separated the tissue into the anterior and the posterior parts. And as you can see here, clearly the posterior presemitic mesoderm is responsible for most of the elongation. This is quantified here, so 48% elongation for the posterior presemitic mesoderm compared to 26% for the anterior, which tells us that as predicted by our earlier model, the posterior presemitic mesoderm appears to be the main component of the triggering elongation in this context. And, and so again, he looked at the effect of blocking FGF signaling. And so I showed you that so these are with the comparing anterior and posterior. So if you look now at the effect of PDO3 inhibiting FGF signaling on the posterior elongation, you see this is a control, and this is the posterior explant treated with 2 different doses. You see you have a very strong inhibition of elongation caused by this inhibition of FGF signaling, which supports the idea that in the FGF signaling triggers this elongation behavior in the posterior part of the presemitic mesoderm. So then we wanted to try to characterize further the system, and particularly to show and measure the forces produced by this elongation triggered by FGF signaling in the presemitic mesoderm. And so what Arthur did is to design a system of cantilever, where he took a very thin glass rod and hooked it to a small piece of aluminum foil. And then he measured the stiffness of these cantilevers. And he developed a system where he could image embryos on the stage of a microscope with the cantilever in place. And he inserted the cantilever at different positions to measure the force generated by the push of the embryo. And so this is just an illustration, simple illustration of the system. You see that the cantilever comes at the posterior end of the embryo. The embryo is covered by mineral oil to limit evaporation. And what you analyze is the deflection of the cantilever, and you know its stiffness, so you can calculate the force which is produced. And so this is so he designed cantilevers of different stiffness. And for instance, if you use one with a low stiffness, like this is 3 millinewtons, you see that so this is the cantilever here. This is the track of the cantilever in red. It's just a dot marking the end of the embryo. You see that the cantilever is pushed in these conditions. But now, if you use a cantilever with a higher stiffness, like 35 millinewton, then you see that elongation is stalled. And so using this approach, you can measure the stalling force, which is necessary to arrest the elongation of the embryo. And so this is just a quantification of the distance measured with these 2 types of cantilever, either soft or stiffer. And so this is a bit work in progress. So I'll come back in the very end to this force measurement. But now I just want to switch gear a bit and try to tell you about another aspect of this story, which is that if what I've told you so far explains elongation of the presemitic mesoderm and possibly of the embryo on a short time scale, but if you want to sustain the entire elongation of the embryonic axis, what you need to do is to constantly inject cells which are loaded with FGF8 in order to sustain this high motility posteriorly, because as I told you, the content of FGF8 is going to decay as it's not transcribed by these cells. So this injection of fuel, if you want, is critical to sustain elongation. And so what, we understand the elongation, but what we did the injection, but we did not understand how this process gets coordinated to the elongation triggered by the presemitic mesoderm and how also it fits with the elongation of the axial organs. And so this has been investigated by Fengshu Xiong, a postdoc in the lab. And what Fengshu did is first to reproduce the experiments that Bertrand did, this ablation of the posterior presemitic mesoderm. And what he saw is that it stores the elongation, as I told you, but also he noted that in this context, the axial organs did not converge as in normal embryos. And so if you measure the distance between the two sides of the neural tube in this ablated embryo, you see that compared to control, it's always higher both for the neural tube and the notochord, suggesting maybe that the paraxial mesoderm is actually not only pushing the system posteriorly, but also compressing the axial organ medially. And so to test that, what he did is to perform ablations and then replace the tissue by very soft alginate gels. And then what he observed is that in this context, the gels get compressed or change shape in a way which is consistent with these compression forces, these mediolateral compression forces. And so this is just a quantification of this, which shows how the aspect ratio is changing in these gel explants. So what this argues is that the paraxial mesoderm not only pushes, as I told you, but also compresses the axial organs mediolaterally. And then a prediction of this behavior is that this compression forces might produce a squeezing, which will act on the growth zone that I talked to you earlier on, and maybe result in this posterior displacement of the growth zone, which is necessary in order to sustain the elongation movements. And then he tested this idea using the same approach, so that is to graft an alginate gel in place of the posterior growth zone. And what he sees is that there is a very different behavior from when you put the alginate gel a bit higher, because here what you see is that you have this indentation into the gel that suggests a posterior push of the axial organs into the terminal growth zone. And so what this experiment suggests now is that the posterior compression results in a squeezing of the axial organs that will push and lead to the posterior displacement of the regions of the progenitors, ensuring the coordination with the forming paraxial mesoderm. And so then what Fengshu did is to establish a clever design to test the role of the posterior force which is produced, which pushes on the posterior domain, and see whether it controls also the immigration of cells. So what he did is to perform an ablation of the posterior axial organs and then replace them by a pin here, a metal pin that he inserted in the embryo in place of the posterior axial organs. And this pin, he could act on it with a magnet in order to bend it, and to apply posterior force on these cells. And so this is the result of these experiments. So this is just at the time of implantation, and this is after implantation. You see that the posterior region has been pushed as a result of the force exerted by the pin. And when he labels cells posterior to the pin in this experiment, what he found is that when you apply a force, what you see is a dispersion of the presemitic mesoderm precursors that are injected in the area. And you can also quantify this. And so the conclusion of these experiments is that the cell diffusion gradient that I discussed earlier not only is involved in generating a posterior push, but also this mediolateral compression that triggers the posterior displacement, the squeezing of the axial organs and the posterior displacement of this progenitor domain, which in turn is going to promote the exit of the cells, and which are then in turn going to generate the posterior push. So in this way, the system establishes a closed positive mechanical feedback loop, which can explain how the posterior elongation movements are sustained in the embryo. And I just want to show you again this movie just to place things in context, this is our posterior growth zone, these are the very highly motile cells which are produced. The idea is that they here exert a posterior push, and this results in the posterior displacement of this tissue here, which further stimulates the immigration of the cells here. And just last 2 slides as a teaser, I want to show you some improvements that were or a new system that was developed in collaboration with Madeleine's group by Feng Shu and Chonyu Chan. And so what they've done is to set up a system on the stage of a microscope where they hooked a chip that harbors AFM cantilevers to a piezo. And so in this way, they can both apply forces on the tissue of a defined magnitude. They can also measure the forces by measuring the deflection of the cantilever, and they can also they're also working at measuring the rheological properties of the tissue using a technique a bit similar to what Susan Eaton described, where it's called chirping, where they measure the oscillations of the probe. And so this is the way it looks like so they started to apply it to measure the force produced by the presemitic mesoderm on the axial organs. That's what you see here. It's actually not very easy to see, but this is the cantilever, the tip of the cantilever, which is at the boundary of the neurotube, and you can measure the force which is produced, which is in the order of hundreds nanonewtons in this case. And this is just an example of applying a force to promote, so that would be to substitute to the pin experiment I discussed. Here, you apply a force of 200 newtons that drags the embryo, so the cantilever is this red tip here. So I'll stop here, just want to acknowledge the people. So this is Feng Shu, and so Feng Shu is now starting his own lab at the Gurdon Institute in Cambridge, and Arthur Michaud did the earlier work I discussed, who's moving to Jérôme's lab this year. So thank you, and I'll stop here.